In order to further understand the histological structures and functions of the lymphatic organs, we should learn the immunocytes and lymphatic tissues firstly. Immunocytes. Lymphocytes include T cells, B cells, and NK cells. T cells. T cells originate in the bone marrow and migrate to the thymus, where they further mature and differentiate and are carried by the blood to other lymphatic tissues. So they are also called thymus-dependent lymphocytes. The newly formed cells are called naive T cells. When they are stimulated by the antigen peptide, most are converted to effector T cells through transformation, proliferation, and differentiation. They attend the cellular immune response. A small part of converted to memory T cells, which keep immunological memory for a long time. Effector T cells are divided into three subpopulations. Cytotoxic T cells, TC cell, act against foreign cells or virus infected cells by means of two mechanisms. TC cells produce perforins that create holes in the membrane, with consequent cell lysis. They also release granzyme through the holes to induce apoptosis. They are like soldiers with knives to kill the enemies. Helper T cells, TH cells, help TC and B cells for immune response by cell factors. The cells are like commanders to control the battle. Suppressor T cells, TS cells, reduce the activity of T and B cells, especially at the end of the immune response, prevent the immune response from overreaction. B cells. B cells originate and mature in the bone marrow and are carried by blood to the secondary lymphoid structures, also called bone marrow-dependent lymphocyte. Effector B cells are plasma cells and take part in humoral immune response by producing antibodies. Effector B cells are like soldiers with guns to kill the enemies. Bullets are the antibodies, so the range of humoral immune response is wider than cellular immune response. Vaccination is to get more memory T cells and B cells. NK cells, natural killer lymphocytes. They attack virus-infected cells, transplanted cells, and cancer cells directly. Mononuclear phagocyte system, MPS. It is composed of the cells that are derived from monocytes in blood. These cells have high capability to phagocytose. MPS includes macrophages, osteoclasts, microglia, Kupffer cells, and dust cells, etc. Antigen-presenting cells, APCS. APCS include macrophages and dendritic cells, DC. They capture and process antigens for presentation to T cells. The second part of immune system is lymphatic tissues. Lymphatic tissue is composed of reticular tissue and lymphocytes and some free cells. Lymphatic tissues are divided into two types based on the composition and appearance: diffuse lymphatic tissue and lymphatic nodule. Diffuse lymphatic tissue mainly contains T cells. has obscure boundary with surrounding connective tissue, has postcapillary venules, high endothelial venules HEVs, which are the important channels for lymphocytes from blood into lymphatic tissue. The circle shows diffuse lymphatic tissue. The left is the pattern diagram of diffuse lymphatic tissue. It shows the high endothelial cells. The square in the right shows a postcapillary venue. Lymphatic nodule is a ball-like structure with distinct boundary. Mainly contains B cells. It includes primary lymphatic nodule, contains small lymphocytes. Secondary lymphatic nodule derived from primary lymphatic nodule with the stimulation by antigens. It has a germinal center. The arrows show lymphatic nodules, the secondary lymphatic nodule. This picture shows a secondary lymphatic nodule with a germinal center. The germinal center includes dark zone and light zone. Naive B cells and memory B cells are stimulated by antigens and move to the primary lymphatic nodule and become larger to form the dark zone. The daughter cells get smaller and more upward to form light zone. The daughter cells, including immature plasma cells and memory B cells, are pushed to the periphery to form the cap. Then the immature plasma cells leave the lymphatic nodule to other places and convert mature plasma cells.